I'd like to welcome Phil and all of you to the live question and answer session of the seminar. Please ask your questions in the box on the screen and we will ask the most popular questions. So if there's one you'd like to see answered, please use the upvoting function. Thank you so much for your talk, Phil. And the very first question is quite a general question, asking whether with genomic medicine, we're in danger of diagnosing cancer when actually what we're spotting is accumulation of mutations.
I think that's a really interesting and very good question. So people are now using the latest technology to find mutant DNA in blood or in urine or in other to sample mutations across the body, and potentially those might be able to be linked to cancer. I think what's interesting about the mutations that we've worked on to date, and it's a very rapidly expanding field, so I can't talk about everything, but I think our mutant cells are unlikely to be releasing vast amounts of DNA into the circulation. They seem to be very well adapted to living in a normal tissue without any inflammation, and actually there's very little cell death in cells in the tissues we work on. But I think in general terms, that is a danger. If you have a background rate of cancer-associated mutations in normal tissues and a very sensitive technology, like some of that are being developed at the moment, there is a potential risk that you could end up, maybe not in people who are normal, but people, say, who have an inflammatory disease affecting some part of the body, and then you could have some cell damage and cell death, and that would release some of the mutant cell DNA, and you get a false positive reading of cancer. So I think the early excitement about these circulating DNA technologies needs to be tempered, because without quite large, rigorous studies on more general populations, we're not going to know whether they're both sensitive and specific enough.
Thank you, that's really helpful.
Okay, Phil, I'll just ask a quick bit more about following on from Julia's question, and I have a kind of second question. Just, is the spectrum of variants that are seen, I guess, in somatic normal tissue, the somatic variants you see, the same as you see in the cancer? So is there any distinguishing feature there, or it is literally the same set of mutations that you're seeing that occur normally as what you would see in a full-blown cancer?
So I think it's a subset of mutations that we see. So I'll speak about what I know, which is the skin and esophagus. We have a very large number of mutations that we've catalogued in those two tissues. Even in what's published, there are probably 10,000, but actually we've got 10 times more than that now that we've accumulated. And the distribution of mutations looks very similar to what's been reported in cancers, with the health warning: there are far too few cancer mutations. So the big genome sequencing projects might have sequenced 100 or a few hundred cancers. And if you spread out all the mutations in the different genes across those, it isn't a large enough sample to answer that question. I have to say, if you look more generally at which domains of which proteins are mutated, it's very similar. So for example, for the gene we've got most mutations for is Notch1, where we probably have upwards of 10,000 missense mutations, never mind the other ones, and that looks very similar. But I think we'll need more detailed mapping of tumor mutations at a much larger scale, as is going on in some projects like Genomics England in the UK, and some other consortia worldwide are scaling this up before we can really answer that question. I think the interesting thing is, so yes, the mutations are very similar, but it's what's the difference between normal and a cancer? And so, for example, in the esophagus, well, pretty much all the tumors have a P53 mutation, but you very rarely, for example, see CDKN1A loss in normal tissue. The mutational signatures of the tumors are dominated by this Apobek process, which again, we have yet to see in the normal tissue. The number of mutations per megabase, the mutational burden, is 1/10 of what is a normal tissue compared to the tumor. And very strikingly, Notch1, which is a fantastic colonizer of normal esophagus. So by the time you're middle-aged, half of or more of your esophagus is null for Notch1 because you've got mutation of one allele and you've lost the other allele. So we're all the oldies are wandering around with very little Notch1 in their esophagus, but that might be good because Notch1's really quite rarely mutated in cancers of the esophagus, might be protective. So this comparison of not so much the individual nucleotide variants, but actually the domains and the relative prevalence of genes in normal and cancer can be very informative, I think.
Fascinating. So I'll ask my question, but I'm going to change the one I was planning to a different one, just because I wanted to follow up on the Notch1, because you mentioned it specifically. Because you have this kind of protective effect, as you mentioned, so you get it, mutations in your normal tissue, but you don't see it so much in cancer. And the question is the mechanism by which Notch1 loss of function mutations are protective understood? And is this simply a fitness effect?
Well, fitness is everything. So in the tissues we work on, they're sort of open plans. So a clone can spread a very long way. I mean, we see clones extending in humans by 1 centimeter. The clones get big and they collide, and then the fittest will outcompete the less fit. And eventually, I guess by the time we're middle-aged or whatever, the fitness landscape is relatively even. So the clones might carry different mutations, but competitively they're about equivalent in terms of fitness. How does Notch1 protect against cancer? Well, we have a couple of ideas which aren't published yet, but I think the evidence is quite mature. So the first thing is we've looked for these microtumors that we find in mouse models, tiny, tiny tumors, 10 or 20 cells. And it seems likely that the very fittest genes in the normal tissue can get rid of those. If you're going to be a baby tumor, you have to outcompete these big, aggressive, very fit clones next door. And the most aggressive clonal colonizer is Notch1. So the idea is that a Notch1 clone can sweep tiny tumors out of the tissue. Whether that's a good thing, a bad thing, or just a thing, I'm not sure, because I think that process of selection eliminates all but tumors carrying strong oncogenic drivers, but I think it's there. Another piece of evidence, which we've done both with genetics by deleting the Notch1 gene in the mouse esophagus and then giving a cigarette-derived chemical to induce tumors and seeing how they grow. Well, when you do that, the tumors are smaller than if you do them with Notch1 wild-type background. But I think more impressively, when you take established lesions, so these are big enough to see, and they're not full-blown cancers, but they're high-grade dysplasias, which are the next best thing. And when you treat those in the mouse with an antibody we got from Genentech in the US, which blocks Notch1 function, and you give the antibody for 6 weeks, the tumors end up smaller. So I think you need the wild-type Notch1 to grow better as a tumor. And this emphasizes the fact that the tumor is not normal, obviously, but the tumor is a different ecosystem, it's a different niche, it's a different competitive environment. So a mutation that does really badly in normal tissue, that would be, or a wild type Notch1 that does really badly in normal tissue can do well in a tumor and be selected for in a tumor. And so I think we're starting to get an insight on that, but don't ask me more than that because I really don't know.
Fascinating. Thank you, Phil.
Thank you. So I wonder if you can expand on your clinical implications of your work. And there is a question if you envisage a day when we actively treat precancer through removing mutant clones in tissue.
I absolutely would love to see that day when we can say these are the bad mutations. And for me, P53 is the standout bad mutation. Okay, that's not a surprise after generations of cancer researchers' work. And certainly in the esophagus, and I think probably in the skin, it's the case that many tumors evolve from P53 mutant cells in normal. And it seems reasonable doesn't mean it's true, but it seems reasonable to speculate that the more p53 mutant cells you have, the greater the risk that they will pick up the winning lottery ticket of genomic rearrangements that will result in a tumor. So reducing the population would be great. Do I think that's feasible? Well, we've done it in a mouse, as I spoke about, with a method that is completely impractical for use in humans. So low-dose ionizing radiation, a couple of CT scans worth, does induce oxidative stress in cells, and that makes, in that context of treating with that radiation dose, p53 mutant cells are fitter and outcompete wild type. Add an antioxidant in, and you reverse the fitness balance, so the wild type is fitter than the mutant. Now, that is a totally impractical, please don't try this at home, wrong thing to do if you want to get rid of your p53 mutant clones, because both antioxidants and radiation have serious health risks. I'm not suggesting that, but in principle, it argues that it may be possible to tilt the fitness balance in a normal tissue to the point where wild type cells outcompete the mutant. So we're exploring this at the moment with whole genome CRISPR screens, gene editing all the genes in the genome and looking at some of the early markers of cells that predict whether cells will be shed from a tissue. So these are differentiation proteins. Maybe we'll find an answer that way. We hope we will. But I would love to see a day where, maybe the oldies and crumblies like me can get a magic pill that will cut my cancer incidence risk so that, I develop my cancer when I'm 110 and have died of something else.
Thanks, Phil. That relates to the next question that's come up about whether the somatic mutation rates in normal tissues increase linearly with age, so they increase over time. And does that correspond with cancer incidence rates with age? So in the esophagus, at least in the small sample that we've published, there's a very strong linear correlation of mutation burden with age. And the mutational signatures that underlie those mutations were signatures 1 and 5, which earlier work by Mike Stratton and colleagues has shown correlate with the age at which a cancer is diagnosed. And the thinking, which is slightly hand-waving, at least in terms of one of those signatures, is that they're cell-intrinsic processes and you're generating mutations just by cells turning over by the passage of time. And that explains that. Now, the incidence of cancer is not linearly related to age. I mean, for most cancers, there's a very strong kind of power law increase. So your incidence pretty low till you get to 60, 65, and then it shoots up. And certainly for the cancers in the tissues we work in, they are diseases of old age. We all remember the young people who get cancer and die off from them, but actually by far the greatest number of things is cases is in old age. Why is that? Well, partly it's because I think the risk of cancer, as I hinted at earlier, depends on the population of mutant clones in the tissue. And a very nice example of this comes from the skin. And this is really developing ideas from decades ago, published by Doug Brash, who's at Yale. But he sort of pointed out that UV light makes mutations in the skin, and we've confirmed that. We have lots and lots of mutations in the skin. But he wondered whether UV light could be expanding p53 mutant clones. And he did an experiment in mice 20 years ago now that seemed to confirm that. And we've revisited that more recently with kind of more upmarket transgenic technology. And it's true, you can show that sub-sunburn doses of UV light, so going to the shops on a Sunday afternoon sort of doses of UV light, will expand pre-existing p53 mutant clones. And that gives you a strongly supralinear rise in the population of p53 mutant cells, and you're inducing new mutations. So I think it's not just about mutation burden, but it's about the interaction of environment, mutation, selection, and time
that's what really is explaining that incidence, perhaps.
Thank you. Thank you.
So, Phil, in your esophagus model, you mentioned that the immune cells didn't seem to have a role in removing these micro tumors. So I guess, you know, do you understand what's happening there? Because clearly immune cells do have a role in tumor cell clearance. And so is that result surprising? Do you know what is happening, for example, in the immune cell population?
I do not want to, these are microtumors, okay? These are the very earliest antecedents, perhaps, of tumors, and 90% of them end up going nowhere, we think, because they're being cleared out by clonal competition. But it's obvious, and certainly in the tumors we work on, the squamous cancers in many sites in the body have a strong component of inflammation. And for example, in the lesions that I talk about being treated with a Notch1 antibody, those have a high inflammatory cell infiltrate. Okay. So I think this, whereas the micro lesions are quite good at inducing angiogenesis, very early, actually, much earlier than sort of the classic Judah Folkman model would suggest, they're not triggering inflammation. But if we look later on, we do see a strong role for the immune system. And I think that you need to look at the kind of the preschool, if you like, stage of tumors differently from the adult cancers. They have a different set of rules, possibly with regard to the immune system. In more general terms, it's very striking that you can have a colossal burden of cancer driver mutations in your tissue that are not edited out. I mean, how is it that half of my face is carrying P53 mutants right now, but I'm not an inflamed mess of immune cells dealing with that?
Yeah. And you don't see the immune cells there. I mean, you're not getting an immune response, or is it somehow suppressed? With the microtumors.
Yeah, in the microtumors.
Yeah, in the microtumors.
So in the microtumors, what we did was we did it two ways. So we took we looked at the clearance rates of the generation and clearance rates of these tumors in these NOD-SCID interleukin receptor mutants, these NSG mice, which are very immune compromised. Yeah. And there's no effect in the NSG rates, and the normal rates are the same. But we also did an imaging with just a marker of leukocytes and showed that statistically with large scale imaging that there's no clustering of leukocytes around lesions at all at this stage. Fast forward later on and add a promoter that drives these into dysplasias, there is a strong correlation, but not at this very early stage.
Interesting. Thank you.
Yeah, there is another question about if these mutations also happens at the same rate in other mammals, and what can we learn about cancer and aging from these?
So they're better qualified people than me to talk about that in our institute. It's very clear, and there's a lovely paper by Inigo Martin-Carino et al. In Nature quite recently, using a new technology to measure mutation burden rates and trying to link that with cancer. I think there's beautiful research going on. I wouldn't want to presume to talk about it. I know that the limited study we've done, we do find, so we worked with the vet school with a pathologist who was doing postmortems on dog esophaguses from dogs that died for other reasons. And we looked at those, and we do find some mutations, but not very many. We tried the same with a horse pathologist from Newmarket, where there's a big racing stables that's quite close to us. And again, we found very few mutations. We didn't follow that up. I think other people at the Institute are following that up using better technologies. I'd be astonished if we weren't all accumulating mutations, but across the animal kingdom, I mean, but it's very difficult doing cancer research in other species, particularly in wild animals, because they tend to just die and nobody knows why. And many of them will die from other causes before they get cancer. And I think there I will stop. So I'll say you will find them if you look for them, I'm sure, but I will not venture outside my area of competence.
Thank you.
Thanks, Phil. I think we can squeeze in probably another question before the top of the hour. And I suppose that one of the questions here has been, what's the implication of your work with respect to healthy aging? Obviously, that's a big push from the UK government at the minute about, aging healthily and what we can do that. So can we potentially enrich for the fitter cells, or is it too early?
I think it is early, but I think, for example, we've shown that one of the cancer-causing mutants, PI3 kinase, does better in diabetic animals, and it affects signaling. And it might be that it'd be very interesting to look at, for example, at obesity and the prevalence of these mutations. Very interesting to look at smoking, not just the mutations being caused by smoking, but the effect that has on the growth of clones. And I hope that we will get to a point where we can start making the lifestyle changes ourselves that will slow the rate of these. I think they're inevitable. We're all going to get them. I just look in the mirror at my skin. I can see the mutant clones there almost with the eye of faith. I think that's clear, but we might be able, it might be that we can tweak the odds of these bad clones colonizing our tissues with some lifestyle interventions.
Thank you.
Great, Phil. So unfortunately, the time is up. It always seems to go very quickly, but that was really fascinating. So thank you so much for your presentation and taking the time to answer the Q&A session today, as well as the audience who has joined us. Just a note to say that actually next month's seminar will actually be given by Roser Vento, the co-host here from our Cellular Genetics program. So we'll be announcing that, but that will be on the 16th of June. And also just a reminder that all the talks and seminars and question and answers are archived on the Sanger Seminar Series web portal, so they can be viewed again, or you can share them with your friends and colleagues. So with that, I'd just like to thank Phil again, as well as my co-chairs, and I hope to see you next month. Thank you.